Fluorescence-lifetime imaging microscopy or FLIM is an imaging technique for producing an image based on the differences in the exponential decay rate of the fluorescence from a fluorescent sample. It can be used as an imaging technique in confocal microscopy, two-photon excitation microscopy, and multiphoton tomography. The lifetime of the fluorophore signal, rather than its intensity, is used to create the image in FLIM. This has the advantage of minimizing the effect of photon scattering in thick layers of sample. Fluorescence lifetimes, a fluorophore which is excited by a photon will drop to the ground state with a certain probability based on the decay rates through a number of different decay pathways. To observe fluorescence, one of these pathways must be by spontaneous emission of a photon. In the ensemble description, the fluorescence emitted will decay with time according to where. In the above, is time, is the fluorescence lifetime, is the initial fluorescence at, and are the rates for each decay pathway, at least one of which must be the fluorescence decay rate. More importantly, the lifetime is independent of the initial intensity and of the emitted light. This can be utilized for making non-intensity based measurements in chemical sensing. Measurement. Fluorescence lifetime imaging yields images with the intensity of each pixel determined by, which allows one to view contrast between materials with different fluorescence decay rates, and also produces images which show changes in other decay pathways, such as infrared imaging. Pulsed illumination equals, fluorescence lifetimes can be determined in the time domain by using a pulsed source. When a population of fluorophores is excited by an ultrashort or delta pulse of light, the time-resolved fluorescence will decay exponentially as described above. However, if the excitation pulse or detection response is wide, the measured fluorescence, dt, will not be purely exponential. The instrumental response function, IRF, t, will be convolved or blended with the decay function, f(t). The instrumental response of the source, detector, and electronics can be measured, usually from scattered excitation light. Recovering the decay function poses additional challenges as division in the frequency domain tends to produce high noise when the denominator is close to zero. TCSPC, time-correlated single-photon counting is usually employed because variations in source intensity and photoelectron amplitudes are ignored. The time resolution can be upwards of 4 picoseconds, and the data obeys Poisson statistics. More specifically, TCSPC records times at which individual photons are detected by something like a photomultiplier tube or an avalanche photodiode after a single pulse. The recordings are repeated for additional pulses, and after enough recorded events, one is able to build a histogram of the number of events across all of these recorded time points. This histogram can then be fit to an exponential function that contains the exponential lifetime decay function of interest, and the lifetime parameter can accordingly be extracted. 16 to 64 multi-channel PMT systems have been commercially available, whereas the recently demonstrated CMOS single-photon avalanche diode TCSPC FLIM systems can offer additional low-cost options. Gating method. Pulse excitation is still used in this method. Before the pulse reaches the sample, some of the light is reflected by a dichroic mirror and gets detected by a photodiode that activates a delay generator controlling a gated optical intensifier that sits in front of the CCD detector. The GOI only allows for detection for the fraction of time when it is open after the delay. Thus, with an adjustable delay generator, one is able to collect fluorescence emission after multiple delay times encompassing the time range of the fluorescence decay of the sample. In recent years, integrated intensified CCD cameras entered the market. These cameras consist of an image intensifier, CCD sensor and an integrated delay generator. ICCD cameras with shortest gating times of down to 200 picoseconds and delay steps of 10 picoseconds allow sub-nanosecond resolution FLIM. In combination with an endoscope, this technique is used for intraoperative diagnosis of brain tumors. Phase modulation equals, fluorescence lifetimes can be determined in the frequency domain by a phase-modulated method. The method uses a light source that is pulsed or modulated at high frequency such as an LED, diode laser or a continuous wave source combined with an acousto-optic modulator, which will modulate the fluorescence, and a modulated detector such as an intensified camera. Since the excited state has a lifetime, the fluorescence will be delayed with respect to the excitation signal, and the lifetime can be determined from the phase shift. Also, Y components to the excitation and fluorescent sine waves will be modulated, and lifetime can be determined from the modulation ratio of these Y components. Hence, two values for the lifetime can be determined from the phase modulation method. Consequently, if the lifetimes that are extracted from the Y component and the phase do not match, it means that you have more than one lifetime species in your sample. An advantage of camera-based frequency domain FLIM is its fast lifetime image acquisition, making it suitable for applications such as live cell research. Analysis. The goal of the analysis algorithm is to extract the pure decay curve from the measured decay and to estimate the lifetime, S. The latter is usually accomplished by fitting single or multi-exponential functions. A variety of methods have been developed to solve this problem. The most widely used technique is the least square iterative reconvolution, which is based on the minimization of the weighted sum of the residuals. In this technique, theoretical exponential decay curves are convoluted with the instrument response function, which is measured separately, and the best fit is found by iterative calculation of the residuals for different inputs until a minimum is found. For a set of observations of the fluorescent signal in time bin I, the lifetime estimation is carried out by minimization of. Besides experimental difficulties, including the wavelength-dependent instrument response function, mathematical treatment of the iterative deconvolution problem is not straightforward, and it is a slow process which in the early days of FLIM made it impractical for a pixel-by-pixel analysis. Non-fitting methods are attractive because they offer a very fast solution to lifetime estimation. One of the major and straightforward techniques in this category is the rapid lifetime determination method. RLD calculates the lifetimes and their amplitudes directly by dividing the decay curve into two parts of equal width t. The analysis is performed by integrating the decay curve in equal time intervals t. E is the recorded signal in the ITH channel and K is the number of channels. The lifetime can be estimated using. For multi-exponential decays, this equation provides the average lifetime. This method can be extended to analyze the exponential decays. One major drawback of this method is that it cannot take into account the instrument response effect, and for this reason the early part of the measured decay curves should be ignored in the analyses. This means that part of the signal is discarded and the accuracy for estimating short lifetimes goes down. One of the interesting features of the convolution theorem is that the integral of the convolution is the product of the factors that make up the integral. There are a few techniques which work in transformed space that exploit this property to recover the pure decay curve from the measured curve. Laplace and Fourier transformation along with Laguerre-Gauss expansion have been used to estimate the lifetime in transformed space. These approaches are faster than the deconvolution-based methods, but they suffer from truncation and sampling problems. Moreover, application of methods like Laguerre-Gauss expansion is mathematically complicated. In Fourier methods, the lifetime of a single exponential decay curve is given by where and n is the harmonic number and t is the total time range of detection. Applications. FLIM has primarily been used in biology as a method to detect photosensitizers in cells and tumors, as well as FRET in instances where radiometric imaging is difficult. The technique was developed in the late 1980s and early 1990s before being more widely applied in the late 1990s. In cell culture, it has been used to study EGF receptor signaling and ERBB1 receptor trafficking. Time domain FLIM has also been used to show the interaction of both types of nuclear intermediate filament proteins lamins A and B1 in distinct homopolymers at the nuclear envelope, which further interact with each other in higher-order structures. FLIM imaging is particularly useful in neurons, where light scattering by brain tissue is problematic for radiometric imaging. In neurons, FLIM imaging using pulsed illumination has been used to study RAS, CAM-CUR, RAC, and RAN family proteins. FLIM has been used in clinical multiphoton tomography to detect intradermal cancer cells as well as pharmaceutical and cosmetic compounds. More recently, FLIM has also been used to detect flavonols in plant cells. FRET imaging since the fluorescence lifetime of a fluorophore depends on both radiative and non-radiative processes, energy transfer from the donor molecule to the acceptor molecule will decrease the lifetime of the donor. Thus, FRET measurements using FLIM can provide a method to discriminate between the state's environments of the fluorophore. In contrast to intensity-based FRET measurements, the FLIM-based FRET measurements are also insensitive to the concentration of fluorophores and can thus filter out artifacts introduced by variations in the concentration and emission intensity across the sample. Dyes and fluorophores for FLIM abbreviations, AB absorption wavelength maximum in nanometers, M, emission wavelength maximum in nanometers, MW, molecular weight, QY, quantum yield, FLT, fluorescence lifetime in nanoseconds, dP, dye-to-protein ratio. See also, Vaser approach to fluorescence lifetime and spectral imaging. References, Joseph Alakowicz. Principles of Fluorescence Spectroscopy 3rd edition. Springer. ISBN 978-0387-31278-1. Lee, Day A. Alt, Jochen. Richardson, Justin. Walker, Richard. Butts, Alex. Stopper, David. Charbon, Bidudo. Henderson, Robert. Real-time fluorescence lifetime imaging system with a 32A, 3201 MCMOS low dark count single photon avalanche diode array. Optics Express 18, 10257 €69. Bibcode, 2010 OEXP 1810257 LDOI, 10.1364/OE. 18.010257. PMID 20588879. Chang, C.W. Sud, D. Misek, M.A. Fluorescence lifetime imaging microscopy. Methods in cell biology. Methods in cell biology 81, 495 €524 doi, 10.1016 per second 0091-679X, 06, 81024-1. ISBN 9780123740250. PMID 17519182. Elson, D.S. Munro, I. Requajo Isidro, J. McGinty, J. Dansby, C. Gallatley, N. Stamp, G.W. Neal, M.A.A. Lever, M.J. Kellett, P.A. Dymoke Bradshaw, A. Hares, J. French, PMW. Real-time time-domain fluorescence lifetime imaging including single-shot acquisition with a segmented optical image intensifier. New Journal of Physics 6, 180. Bibcode, 2004 NJPH, 6180 EDOI, 10.10886180. Sun, Yinghua. Hatami, Nisa. Yi, Matthew. Marcoux, Jennifer. Elson, Daniel S. Gorin, Frederick. Schrott, Rudolph J. Phipps, Laura. Fluorescence lifetime imaging microscopy for brain tumor image-guided surgery. Journal of Biomedical Optics 15. Bibcode, 2010 JBO, 15E6022 SDOI, 10.1117/1.34866612. Godella, TWJ, Editor, FRET and FLIM Techniques. Elsevier, 2009 http, books Google.com about FRET and FLIM Techniques HTML? It equals UHV-QU4HLHH8C and Reader equals Y, Oida, T. Sacco, Y. Kazumi, A. Fluorescence lifetime imaging microscopy. Methodology development and application to studies of endosome fusion in single cells. Biophysical Journal 64, 676 €85. Bibcode, 1993 BPJ, 64676 doi, 10.1016 per second 0006-3495, 93, 81427-9. PMC 1262380. PMID 8471720. Lakowicz, Joseph R. Smasinski, Henryk. Nowakuk, Kazimierz. Berndt, Klaus W. Johnson, Michael. Fluorescence Lifetime Imaging. Analytical Biochemistry 202, 316 €30 doi, 10.1016/0003-2697, 92, 90112 K. PMID 1519759. Lakowicz, Joseph R. Smasinski, H. Nowakuk, K. Johnson, M. L. Fluorescence Lifetime Imaging of Free and Protein-Bound NADH. Proceedings of the National Academy of Sciences of the United States of America 89, 1271 €5. Bibcode, 1992 PNAS. 89.1271 L doi, 10.1073/FNAS 89.4.1271. PMC 48431. PMID 1741380. Wouters, Fred S. Bayesians, Philippe I. H. Fluorescence Lifetime Imaging of Receptor Tyrosine Kinase Activity in Cells. Current Biology 9, 1127 €30 doi, 10.1016 per second 0960-9822, 99, 80484-9. PMID 10531012. Vervier, Peter J. Wouters, F.S. Reynolds, A.R. Bayesians, P.I. Quantitative imaging of lateral ERBB1 receptor signal propagation in the plasma membrane. Science 290, 1567 €70. Bibcode, 2000 Sci. 290.1567 V doi, 10.1126/Science. 290-5496-1567. PMID 11090353. Delbar, Owen. Tramier, Mark. Copy Moisen, Mortar Copyright. Gaillard, Claire. Corvalin, Jean-Claude. Buendia, Brigitte. The truncated prelaminae in Hutchine-Sauna Guilford progeria syndrome alters segregation of A-type and B-type lamin homopolymers. Human Molecular Genetics 15, 1113 1122. doi, 10.1093 DDL026. Yasuda, Ryohyu. Imaging Spatiotemporal Dynamics of Neuronal Signaling using Fluorescence Resonance Energy Transfer and Fluorescence Lifetime Imaging Microscopy. Current Opinion in Neurobiology 16, 551 €61 doi, 10.1016/JCONB. 2006.08.012. PMID 16971112. Harvey, Christopher D. Yasuda, R. Tsung, H. Svoboda, K. The spread of RAS activity triggered by activation of a single dendritic spine. Science 321, 136 €40. Bibcode, 2008 Sci. 321 136 H doi, 10.1126/Science 1159675. PMC 2745709. PMID 18556515. Calabi, PETR. Sodholm, John. The Design of FAR Paragraph RSTE Resonance Energy Transfer-Based Molecular Sensors for RAN GTPASE. Methods 51, 220 euro 32 doi, 10.1016/JMF. 2010.01.022. PMC 2,884,063. PMID 20,096,786. Mueller-Harvey, Irene. Fucht, Walter. Polster, Jürgen. Tomcover, Lucy. Burgos, Pierre. Parker, Anthony W. Botchway, Stanley W. Two-photon excitation with picosecond fluorescence lifetime imaging to detect nuclear association of flavanols. Analytica Chemical Actor 719, 68 euro 75 doi, 10.1016/JACA. 2011.12.068. PMID 22,340,533. Becker, Wolfgang. Bergman, Axel. Lifetime imaging techniques for optical microscopy. External links: Fluorescence excited state lifetime imaging, lifetime and spectral analysis tools and image J, http://spectron.com.